So this is the first of probably a couple, at least, of introductions to sex-linked genes. So we're going to look at um, the examples we've looked at in the previous lecture. Single gene inheritance were all autosomal traits. That is, that in the organisms studied, the same number of autosomes are present in males and females. And in this lecture, we're going to talk about genes that are involved with sex determination, but we're not going to look at sex determination. It's very interesting and it's very complicated, but we're just going to look at the inheritance of genes on those particular chromosomes, because those chromosomes have slightly different segregation patterns than the ones we've been looking at. And the reason why it's important to know this is more for the case of genetic counseling type situations. So we're not talking about gender or how a person presents themselves, but if we're trying to figure out their likelihood of having a particular mutation, knowing which sex chromosomes they have will help us determine that with proper accuracy. And so that's really the main reason. And so none of this is about gender or gender presentation. This is just really about what we would call chromosomal sex, and it's really because if you're trying to follow alleles through one generation to the next, knowing how they're passed on really helps you with things like probability calculations and that sort of thing. Okay, so these are just two examples. This is a particularly pretty example of an X-linked trait, which I'll talk about towards the end. And these are not real flies, of course, these are schematics, but we're going to be talking about the mutation white, which is a mutation in a gene on the X chromosome of flies. And those are the examples, the main examples we'll go through. I'm including this just for you to have a look at, because I will have in a previous class have asked some questions that just show you some chromosomes and then just ask you to identify whether it's showing meiosis or mitosis, and then some description of how you know. So you might want to take a look at that before you proceed. So autosomes. Sometimes people get a little bit confused about autosomes, and they think that autosomes and sex chromosomes are in different cells. But all your cells have all the same chromosomes except for your gametes, which have only one of each chromosome. And the rest of your cells have two of each chromosome. So basically, when we're talking about autosomes, these are the ones where females have two of each and males have two of each. And they're essentially then comparable sets. And they're referred to as autosomes. And so when we're looking at segregations of the genes on autosomes, we will, when we're talking about reciprocal crosses, which we will do in a moment, and which I did introduce in the last lecture, you should get the same results regardless of sex. So it doesn't matter, for example, if you have a female who's homozygous for a mutation crossed to a male that's maybe wild type for that gene. The little plus superscript means wild type. So if you did the cross this way, you'd get, all the progeny would be heterozygous, whether they were male or female. And if you did the cross the other way, with males that were mutant and females that were wild type, you'd still get the same result, right? All the progeny would have a certain phenotype and genotype. But if these were sex chromosomes, you would have some different results depending on which way the cross goes. And that's one of the key points from today's lecture. So sex chromosomes are a pair that differs in males compared to females. Okay, and the chromosomes, the word for the chromosomes is heteromorphic Hetero means different and morph means form, so different form. And when you physically look at electron micrographs and other things under the microscope, you will actually see different appearance. So the X chromosome replicated and ready for cell division might look like that, whereas the Y chromosome might look more like that, a smaller, more compact, and it has many fewer genes and so on. Now there's quite a few organisms that don't determine sex based on sex chromosomes. Some do it via temperature. There are some organisms where you have a whole school of fish, for example, and there's one male and in charge of the school. And if you take the male out, one of the females can transform into a male and can produce functional sperm within 24 hours or 2 days or something like that. So there's lots of organisms. Slugs are hermaphrodites. They're both male and female. There's organisms that start off as female and as they get older become male and vice versa. There's lots and lots of different ways this happens and lots of variety. We're really only looking at a very limited set, partly because X and Y is how things work in humans, but also because if we're interested in chromosomes and the transmission of alleles and so on, we can actually make connections between a physically different X and Y chromosome and how those genes are being passed on. Okay, but many plants don't have sex chromosomes either. Now, one thing that happens, even though these chromosomes don't look very similar to each other, they have small regions just on the tips that are similar and allow them to pair with each other during meiosis 1 and then separate away from each other during meiosis 1, just as if they were homologs of each other. But it's really important to keep in mind that males in an XY system, which is the one we're starting with, their X chromosome has probably thousands, a couple thousand genes on it. The Y chromosome has a very small number of genes, and they are not the same genes that are on the X chromosome. And we'll talk about that a little bit more. Okay, but they usually the genes are about fertility, about making sperm tails, which are very important if you're a male. But there's not much other similarity to the X chromosomes. Okay, so very important point, and I hope this will help us to head off potential misunderstanding. So all of your cells have the same autosomes, pairs of autosomes, and you have sex chromosomes. So all the cells have all of them. In diploid organisms, your somatic cells, which is most of your cells, have 2 of each chromosome. If you're female, of course, your sex chromosomes will be 2 of the same. And if you're male, you'll have 2 different ones, but you'll still have 2 of those sex chromosomes. In the gametes, you'll have just 1 of each chromosome. Okay, but autosomes, when you make gametes, they have autosomes and sex chromosomes. The rest of your cells have autosomes and sex chromosomes. So they're not divided up amongst your body cells or anything. The same chromosomes, the same genes, and so on are present in all the cells of your body. And these are the important terms. When we talk about the chromosomes, this is a way of describing the sex chromosomes. They are heteromorphic, and heteromorphic means they look different from each other. And they're just 2 systems we're going to be looking at today. And then if we're talking about the sex or the individual, if we're talking about the XY individual or the WZ individual, we're talking about heterogametic. Hetero means different, gamete means gametic, means gamete, and this means that when they make a gamete, the gamete can have either an X or a Y, or the gamete can have either a W or a Z chromosome. So the 2 gametes will be different from each other based on the sex chromosome that gets put in. Homogametic sex, the homogametic individuals, they can only produce gametes with the same sex chromosome. Females in the XY system, all their gametes are going to have an X because that's all the chromosome they have, or with only a Z if they're males in the ZW system. So this is the actual individuals or the sex, whereas this is the actual chromosomes. And so it's important to use those terms correctly. And this is just an example. So it shows you the sex chromosomes of, this could be humans or any mammal, with a couple of exceptions. Females have two X chromosomes that are the same gene, same order, possibly allelic differences. Males have just the one X chromosome, so if there's a recessive mutation, it's going to show because they don't have a wild type copy on the homolog, and then a smaller Y chromosome. Okay, and in birds and other non-avian reptiles, birds are technically reptiles, and so the rest of the reptiles are called non-avian reptiles. In butterflies and moths and some other organisms, there is what is called a ZW system, and it's the exact opposite. So there are two of the same sex chromosomes in males, so they have two Z chromosomes, they're called, and females have two different ones. So in this case, the females are the heterogametic sex, and the males are the homogametic sex. So everything I'm going to talk about the way segregation occurs in the XY system is opposite with regard to the sex of the individuals. But the asymmetry of it is still the same idea. Okay, so we're going to start with this one. This is the one we're familiar with. Almost everybody already knows about, the XY system and a little bit about it. Okay, and oops. So the X and the Y chromosomes determine chromosomal sex. There's a maleness gene on the Y chromosome. It's a transcription factor. It's called SRY, sex determining region on the Y. And just in general terms, there's way more detail and nuance than all this. But in general, this transcription factor on the Y chromosome initiates the transcription of genes all over the genome that cause an embryo to develop as a male. And if the SRY gene is absent, then those genes don't get turned on and development proceeds on a sort of a female pathway. Now, there are special regions that I already mentioned on the very tips of the chromosomes, and these are similar. That's why they're colored the same. Region 1 at one end and region 2 at the other. These are actually very similar in sequence, and there are some similar sequences around the centromeres that allow for these to actually pair with each other. The X chromosome has to coil around a lot so that this part can pair with this part. The centromeres can pair with each other, and these bits can pair with each other during meiosis 1, and then separate from each other. Okay. Otherwise, the sequences in between here, all of this stuff is very different between the two chromosomes. Okay. And so sex chromosome inheritance in humans and other mammals, the female has gametes that all produce eggs with X chromosome. Males, 50% of the sperm have an X, 50% have a Y, and so we get 50% male to female ratio. In fact, sort of interesting, male sperm that have a Y chromosome are a little teeny bit lighter than sperm that have an X chromosome, just a little bit different. And so they swim just a tiny bit faster. So the fertilization rate, if you were to look at fertilized embryos, and if you were to look at live births, the ratio is just slightly more males than females, so 50.5 to 49.5 or something like that. But there are a number of conditions on the X chromosome that are potentially lethal or harmful, and so there are more male babies that have genetic conditions that cause death, which is really very sad. That brings the ratio back down to about 50-50. And there are a number of serious conditions on the X chromosome. If we have time, we can talk about those at some point in the class. And just a reminder that, of course, the autosomes are segregating too, but we're just looking at the sex chromosomes here. When we look at Punnett squares and that sort of thing, we usually only look at one gene at a time or a couple of genes at a time, but the others are all still segregating normally. It's just that we're not following or paying attention to those. But it's not like only sex chromosomes are segregating here. Okay, and so in X-linked inheritance, depending on what crosses are done, the males and females can show different phenotypes, different genotypes, and the ratios in the results of a cross could be different. And I'm going to just, I think, finish off here with just showing you the genetic notation. So for autosomal genes, we were using uppercase to indicate a dominant allele and lowercase to indicate a recessive allele, or commonly the wild type to indicate a functional wild type allele, usually the dominant allele, and then no superscript, or you could have a minus to indicate a mutant allele. These are all normal ways to express a genotype, and I do tend to put the slash in simply because once we're starting to deal with multiple genes, we want to have it in the genotype, so we might as well have it for single genes too. Females, there are three possible for a single gene, three possible X-linked genotypes. So you can be homozygous, you can be heterozygous, or homozygous for the recessive. And just a note that the way we designate the alleles, we indicate that they're X-linked by writing the X chromosome and then just superscripting that this is one of the alleles on that X chromosome. There are, of course, hundreds, thousands probably of other genes, but we're just following one gene in a particular cross. Now, males have two possible genotypes. They can either have a dominant or a recessive allele. Pardon me. Their other chromosome is a Y, and the Y chromosome doesn't have any of the corresponding genes on it. And so we refer to this as hemizygous. Okay, and I will stop there. So this coughing fit will go away, and you can pick it up again on the next recording.